let's start from the beginning. Why we are called Ramazzini Institute? Because Bernardino Ramazzini, around 300 years ago, wrote this wonderful book, De Morbis Scientificum Diatriba, which was the first book of environmental and occupational health, and is considered the pioneer of the field. Therefore, our founder, Cesare Maltoni, decided to entitle to Bernardino Ramazzini our institute. So our institute is a social cooperative, a no-profit social cooperative that aims to do research on primary and secondary prevention on environmental diseases, including cancer, but not only cancer. And we do scientific research, we do training for any level from EU inspectors to high schools in the field of environmental health, and indeed we do also secondary prevention activities, so a clinical activity in our clinical practice in Bologna, in particular for the early detection of tumors. We have an international scientific advisory board that every year overlooks on our activity, including members of the NIHS, the Ministry of Health in Italy, WHO, and different institutions from the US and Europe. And here is the very lucky location of our research center, which is a 15th century castle that was donated for 100 years to Cesare Maltoni by the municipality of Bentivoglio in the province of Bologna to do research for 100 years on environmental chemicals. Here is a little bit of how our castle looks like from a history perspective. I can complain indeed about the quality of the work of a castle. And these are some of our research labs that you see. Some of them are located with frescoes around, but other ones are more modern and actually also more unfortunately we had to readapt the castle to the need, for example, of air circulation, of tightening and sealing that modern lab has. So we had to give up some of the historical part in spite of science to do better science. So here is a brief overview of the compounds that we studied in the last around 50 years of activity of our research center. So it's 210 compounds assessed for carcinogenicity, and we showed clear evidence of carcinogenicity just on around a half of these compounds. So most of them, the majority of them, did not show evidence of carcinogenicity. Unfortunately, most of the time the negative evidence, as you know, is not that well known and that well spread as the bad news. But indeed these results are very important, very, very important for public health decision makers and for regulatory makers in order to decide which products and which chemicals might be safe to use. And a few words on why carcinogenicity bioassays are important and have been long established as the most predictive model for carcinogens in humans, and not only have a role in predicting the carcinogenicity, but also retrospectively assess whether exposure that have shown some epidemiological evidence of carcinogenicity, for example asbestos, for example benzene, where and at what doses were carcinogenic also in experimental models that do not have all the confounding factors that are obviously present in the real life setting, which is the one that occurs all the time in humans. So here a brief overview of some of the substances that we studied and how predictive was our model that is also 99% consistent on with the model that is also used by the National Toxicology Program in the US, which is our counterpart, or better said, we are the counterpart in Italy of what the NTP is. I believe most of you are familiar with the National Toxicology Program, but indeed they are the #1 of in terms of number of compounds tested, with over 900 compounds tested for their carcinogenicity. So here you can see vinyl chloride, benzene, trichloroethylene. These are only some of the substances that we studied, we discovered as a carcinogen. Almost 30 years before, they were evaluated as known carcinogen by the IARC. For the one of you that are familiar with the IARC evaluation, to become a group 1 carcinogen, you need also evidence from epidemiological studies. So the most you can make in terms of evaluation as a carcinogen based on carcinogen bioassays is 2A, probable carcinogen for humans. To become 1, you need also evidence from epidemiological studies. Again, asbestos, formaldehyde are other substances that we studied, and we move forward until diesel exhaust and fluorodenide, which is a asbestiform fiber present in Sicily in the most recent years. So there are a number of challenges that toxicology is now facing that go beyond the standard carcinogenicity bioassay that we always perform. So first of all, as you can see, we have a problem which is lack of funding for independent research, both in Europe but also in the US. And we have another problem which is assessing low doses of multiple chemicals present in the environment, in the general environment, while before we had mainly exposure occurring in the occupational setting. Then we have multiple effects that we have to address. Not only carcinogenicity anymore, but also a number of other endpoints, such as reproductive toxicity, endocrine disruption. We need also to reduce the number of animals, because indeed the regulation in use in both Europe and in the US call for reducing, refining, and replacing the use of animals. Then there is an increased need for specialization and complexity of these studies that was not present in the early days of carcinogenicity and toxicity bioassay. They were, I would say, almost very rough evaluation of mortality, lethal doses. This was what we were assessing before. Now we are assessing microbiome. We are assessing very fine changes that are predictive for the human health effects. So, as you can imagine, the more the specialization, the higher the complexity and the expertise involved in the studies. And then indeed we need to integrate all this evidence with the epidemiological findings and with the mechanistic findings from the in vitro work. The case where we aim to address all these challenges in toxicology, just to make sure that we want to complicate our life sufficiently, was on the glyphosate case. Why the glyphosate case? Because glyphosate is the most sold pesticide in the world. Here you can see the exposure that dramatically increased in the US, particularly connected with the use of genetically modified organisms that are actually designed to be resistant to glyphosate. Why? Because glyphosate is an herbicide, is a very effective herbicide, and when you are exposed to glyphosate, all the plants die, all the infested plants die, and if you have a resistant corn or soybean, then the plant can survive while the other ones are killed by glyphosate. So it's a very effective compound to be used in agriculture, and for this reason have been increasingly used not only in the GMO setting, but also, for example, in Europe, where GMOs are banned, the level of glyphosate are also exploding in the last 20 years in terms of use because of the effectiveness of the product. We are now talking about over 90% of the agriculture in the US that is based on this GMO glyphosate-resistant product. So it's a very ubiquitous exposure that have been found in rain, water, air, almost in every matrix that was measured. Furthermore, there were discrepancies in the evaluation between WHO and, for example, the European Food Safety Authority, and WHO in particular the IARC, which is the branch that evaluates carcinogens, in terms of the carcinogenicity of this compound. The IARC said it's a probable carcinogen, 2A. EFSA said there's no risk of carcinogenicity. In particular, IARC makes evaluation on hazard, so qualitative evaluation whether a substance is or is not a carcinogen. The European Food Safety Authority mainly makes evaluation of risk, so whether the level of exposure that are actually present in food are increasing the level of cancer, the burden of cancer in humans. Just to give you a previous idea, an idea of the previous evaluation of the IARC, just 5 pesticides between 1971 and 2014 were evaluated as a probable or a known carcinogen by the IARC. Then the evaluation of glyphosate and other 2 organophosphate, because it's very important to remember that IARC evaluated 3 organophosphate, also malathion and diazinon, together with glyphosate, and evaluated them as 2A, so probable carcinogen. So in general, IARC, at least previously, tended to be very conservative in making a call with if a substance was a group 1 or 2A carcinogen. Even more complicated news of yesterday, the PEST committee of the European Union, which was entitled to revision the decision by EFSA to call as a non-carcinogen glyphosate, said that glyphosate carcinogenicity should be reassessed, and there's need for long-term studies, and in general the pesticide risk assessment evaluation in Europe should be changed in order to be more adequate to predict eventual health effects in humans. So as you can see, the topic is extremely hot in both sides of the ocean, going through China, which is the main producer of glyphosate. So there's not a single place now that can be called glyphosate-free, at least to my knowledge. So we embarked in this adventure, as you will see, with many colleagues of different institutions, of the Global Glyphosate Study, which is aiming to address in vivo experiments the effect of glyphosate before starting with a pilot phase, so a smaller experiment that you'll see now how we designed and how was tailored to be predictive for some of the, and interesting to know some of the effects that should be explored in long-term settings, and that aim to mimic the real-life exposure of humans, because many of the assessments that you saw, both of EFSA and of IARC, were based on high level of exposure, doses that are extremely high. Not that this is rare in toxicology, but the exposure levels were around 100 or 170 milligrams per kilogram of body weight. The Global Glyphosate Study started in 2016 and aimed to address all these challenges of toxicology at once, or at least we tried. One was trying to gather funds in more alternative ways. So we are a social cooperative, and the first pilot study was funded by the 30,000 associates that we have. So it was a local but globally affecting study, indeed locally funded by our associates with Crescentina and Tagliatelle and Bolognese sauce events that gave the effect that you also can see. I cannot blame the endocrine disruptors, but also finding new strategies like crowdfunding campaigns. Crowdfunding campaigns that for now already set a record in toxicology with around 130,000 euros that were retrieved through our crowdfunding website, and I'll tell you more about how our strategy of crowdfunding is proceeding. Second, the challenges of low doses, and here we decided to start from scratches with low doses that are comparable to the one that humans are exposed to. In particular, you see the acceptable daily intake that is currently admitted in the US and in Europe are the reference doses for us to start with and to be addressed in our experimental setting. Then we have multiple effects that have to be assessed, and here we aim to address both in the pilot phase that was just concluded, and you'll see the results in a few minutes, and in the long-term setting. Our idea was to address not only carcinogenicity anymore, but also reproductive, endocrine disruptive effect, microbiome effects, and chronic toxicity effects in an all integrated design. This is a brief overview on the endpoints that we just addressed in the pilot study. So the one that we aim to address in the long-term study are even more, but from anogenital distance to sperma, aneuploidy and sperm alterations to hormone alterations up to micronuclei, there are a whole number of endpoints that are investigated in different windows of susceptibility, and we'll see now how important that is to have different windows of susceptibility studied. And indeed, the number of animals aim to be reduced, in particular in the long-term study, using an innovative integrated experimental design that we proposed the scientific community like 4 years ago, and was published in EHP in collaboration with other colleagues also here in the US. And very briefly, the aim of this integrated design is to unify different guidelines currently in use for carcinogenicity, reproductive toxicity, and neurotoxicity in a single experimental setting in order to reduce the number of animals used, optimize the number of endpoints assessed, and also the window susceptibility assessed, and using animals from the same generation that have to be addressed for the different endpoints in different labs. So the different lab in blind will assess the specific endpoints, for example, microbiome alteration or micronuclei alterations, independently from the place where the in vivo experiment occurred, in order to have a better and more multi-center assessment that proved to be efficacious also in the setting of the clinical world, for example. Then, for the increased complexity and specialization, what I would say is just we had to check for many partnerships and for many institutions. One of them is indeed the George Washington University, to address the different endpoints. So this is the members of our team so far in the pilot study, and will be a larger group in the long-term study. From the University of Bologna to the Ministry of Health in Italy, to the Mount Sinai School of Medicine in New York, and the George Washington University, all of them were providing their specific expertise in the different endpoints and in the assessment of the different endpoints. And this, we believe, made our design first and assessment afterwards of the endpoints very, very solid. And last challenge was how to make this all experimental design fruitful and useful for the risk assessment and policymaker expert. So first of all, we tried to identify doses that were relevant, as you already saw, for the human exposure, reduce the sources of bias and make statistical calculation whenever possible of the power needed for the study. And indeed, from the first pilot study that you will see happen at just one dose, so it's a pilot study that cannot address issues like dose response that are fundamental in the risk assessment, try to have an indication of which doses might be useful to be studied, if lower or higher, in the next long-term assessment. And an identical mindset was used for addressing already from the pilot study some outcome, an endpoint that is particularly relevant and and was associated with glyphosate exposure. Here, for example, genotoxicity, that was heavily discussed in connection with exposure to glyphosate, and was addressed with micronuclei from the very first phase of the pilot study at doses that are generally not screened for in micronuclei standard assessment. So here, let's start with the pilot study and giving you a few results of the pilot study that emerged this year. So first of all, in the glyphosate world, the issue that is rarely addressed is what is the role of adjuvants. So most of the glyphosate exposure occurs together with exposure to adjuvants. Almost 700 products just in the US are containing glyphosate, but sometimes up to 15% of the formulation is not glyphosate. These adjuvants that sometimes are known, sometimes are covered by by commercial secret. And whether so the glyphosate or glyphosate with formulation might have different effects, and what are the possible effects that glyphosate might have, except from cancer, that was the most discussed endpoint, but not necessarily the most important, or not necessarily the unique endpoint of interest for glyphosate exposure. So we started this adventure, I would say, in 2016, and we started to address the outcome starting from pregnancy exposure up to young adulthood, so 13 weeks of age in Sprague-Dawley rats. And here a little bit more details. So we had drinking water exposure, controls, glyphosate at US ADI dose, so 1.75 milligram per kilogram of body weight per day. And Roundup exposure, the Roundup formulation that we decided to use was the Roundup Bioflow, because this is the formulation that was used in the reassessment of glyphosate in Europe that happened in 2017, and relicensed glyphosate in Europe until 2022. As you can see, specifically Roundup Bioflow contains 16% of unknown surfactants and adjuvants. So unfortunately, we don't have idea what this substance might be. And the strain that we use, Sprague-Dawley rats, are the one used both at the NTP and at the Ramazzini Institute for carcinogenicity bioassays, for endocrine disruptors assays, and also for toxicological assays starting from 28 and 90 days toxicological bioassays. A little bit more about the design. Exposure started from gestational day 6 And there was a cohort that we studied up until 6 weeks of age after weaning, developmental cohort, and another cohort called toxicity cohort that was studied up until 13 weeks of age. So, indeed, the pilot study could address some issues, not all, because obviously you need multiple dosage studies or long-term studies to address some outcome, as for example chronic toxicity or carcinogenicity. But indeed this could provide some initial evidence of target organs, some initial evidence of possible endocrine disruption activity rather than genotoxicity, and also initial evidence for prenatal exposure, because very few studies addressed the issues connected with exposure to glyphosate, which we know occurs starting from pregnancy. There are epidemiological evidence also in particular coming in from the US, from Indiana cohort exposed pregnant women where low birth weight, statistically significant low birth weight was observed. So here are the results in terms of exposure, because one of the things that who is more familiar with glyphosate knows is how important is glyphosate rather than AMPA as a biomarker is a matter of discussion, because AMPA is the main metabolite of glyphosate, but just a little bit of AMPA is of glyphosate is converted to AMPA. Depending on the species, including humans, is most often less than 1%. So it's a metabolite that might not be always that indicative as glyphosate per se as a biomarker, and our result confirm that glyphosate is indeed present in the urine of exposed rats at levels much higher than AMPA itself. But indeed both AMPA and glyphosate are biomarkers of exposure to glyphosate. What is more important here is to observe that the glyphosate and Roundup, glyphosate and AMPA in urine were comparable. And you see more details now about that, but adjuvants didn't seem to play a major role in the absorption of the chemical, which was so far something that we didn't know whether these adjuvants might play, at least the one that we studied in the biomarker, in the Roundup bioflow. Here is an evidence that was so far not present in the literature, whether glyphosate might bioaccumulate or not in animals. And there was much discussion and speculation about that, but very few studies address this issue. And what we observed, as you can see, is an increasing glyphosate dose in the urine, both in a comparable one in Roundup and glyphosate, but a dose-dependent increase in urine glyphosate depending on exposure time. So this was early evidence of bioaccumulation that we aim to investigate more, but is extremely important knowing that pesticides or substances that tend to accumulate might indeed represent a higher risk if any effect is proven at the doses that humans are exposed to are any relevant for some health outcome. So indeed glyphosate is a most accurate biomarker, at least in experimental models of exposure to glyphosate. Adjuvants didn't seem to play a role, and indeed we aim in the long-term study to assess whether this bioaccumulation occurs also in the long-term setting and with different doses, because it might be that different doses accumulate differently in rats, but also in humans. Here are the effects that we observe on the microbiome, and I will just point out here this red part, which is very interesting for the concept of windows of susceptibility. Indeed, we did not observe alteration of the microbiome in dams at the doses that we studied. Again, a dose that is currently admitted in the US also has exposure for children. But in pups, the same dose give statistically significant alteration, both in terms of reduction and in terms of species heterogeneity in pups, in particular before in the prepubertal age. Furthermore, glyphosate, here you can see that Roundup, more than glyphosate, had a more powerful effect on altering the microbiome. So not only glyphosate was capable of altering the microbiome, also the surfactants present there had some additive effect on glyphosate on disrupting the microbiome, which comes in some sense as no surprise, because glyphosate was patented as an antibiotic and was known to have an effect on possibly on bacteria and parasites. And
excuse me, in the pups, what organ or tissue are you, or sample?
intestinal microbiome.
Intestinal.
And it's
Yes.
you're looking for diversity only, not
Diversity.
functionality.
Not functionality, we were looking for diversity and quantity. Actually, this endpoint was assessed by the Mount Sinai School of Medicine specifically. And these data that I've shown you are described in more detail in the publications that we released this year in Environmental Health, both on the microbiome effect and on the effect on the first endpoints on bioaccumulation, and more will follow and are currently under review on the effects we observe on increased anogenital distance in female and male, in particular exposed to Roundup but also glyphosate, and a statistically significant delay of the first estrus in female. So these two effects are like a masculinization effect, androgenic-like effect, and also a statistically significant increase in micronuclei frequency, both in male and female exposed to Roundup, which is quite remarkable considering that these are low doses and the number of animals for the toxicologists present in the room are low. So allegedly this is a design that is at the edge, if not underpowered, to detect in general micronuclei frequency increase. So a brief summary about what we observed in this pilot study is that glyphosate-based herbicides were able to alter sexual development, genotoxicity, and microbiome parameters already at doses that are admitted in the US. We observed this bioaccumulation effect that was not described before, and we also observed that glyphosate and formulations might have different effects, including for carcinogenicity. So it's extremely important to observe in a long-term setting whether glyphosate or glyphosate formulation have the same effects. So indeed a long-term study is now needed to know in detail and further deepen this initial evidence that we observed in the pilot. Our results were presented before the European Parliament in May this year, and also the PEST committee was present when we presented our results, and we are now trying to gather the funds and the necessary support through institutions and through our crowdfunding campaign that is currently occurring, and you'll find here the website www.glyphosatestudy.org for more details on the campaign. And with this, I thank you so much, and I'm glad to take any questions from you.